Bro, it's really still fucking hot in here.
Kind of. I mean, you studied to do it, is my point. You know the material, so it's valid. So, yeah.
Okay.
Let's get back into the exciting world of the Actin skeleton. A quick recap is to remember actin, the G monomers of actin are present at really high concentrations in the cell. So the amount of G-actin in the cell is way above critical concentration, so there's a number of proteins that are present in the cell also to kind of keep actin from polymerizing, so you have thymosin and profilin and cofilin that kind of play a role together to keep that huge amount of G-actin under control. And then we started talking about other proteins that also do that, and these are proteins called capping proteins that cap both the plus and that can cap either the plus or the minus end. Okay, so actin polymerization is powerful. It can move things, which we'll talk about today. We'll talk about how actin moves a lot of things. Because it can do so, the cell wants to regulate how and when actin polymerizes. And one of the ways it can do that is by capping either the plus end or the minus end, or both ends. Right, so they are capping proteins in the cells. In cells, the two major ones is one called cont'd and tropomodulin. Okay, so you have the plus end capping protein cont'd. Cont'd is present, the plus end cannot polymerize or depolymerize. So anything that happens will be through the minus end, and the minus end has a specific critical concentration. So if you've capped the plus end, then you're dealing with the minus end, and whether it's going to polymerize or depolymerize can be regulated, and things can happen accordingly. Similarly, there's another protein called tropomodulin, which caps the minus end. If you've capped the minus end, it cannot polymerize nor depolymerize. So all activity will be at the plus end. The plus end has its own critical concentration, so 0.12 micromolar. So with this configuration, the plus end can grow and shrink and do things depending on what the cell needs to be done. Okay, of course, you can imagine a situation where you've actually capped both ends. You've capped the plus end with cont'd and the minus end with tropomodulin, and then that actin microfilament will stay that size because it can't polymerize or depolymerize. Okay, so there's proteins that can regulate what's happening at the plus or the minus end or both ends if they're both capped. So that's in the cell. These are natural proteins. In terms of research tools, there's a number of drugs that can also be used to study actin dynamics. For example, there's a drug called cytocolazin. When you add cytocolazin, it depolymerizes actin microfilaments. So if you want to see what actin is doing, you can depolymerize it and then look at repolymerization or sort of other dynamics of what's happening inside the cell. So cytocolazin can depolymerize actin, whereas a chemical named phalloidin stabilizes actin. So by stabilizing, I mean it's not going to depolymerize. Okay, just like with Taxol and microtubules, different doses can do different things. But for our purposes, if you add phalloidin, it's just not going to shrink, so the size of it will basically stay the same, and you can use that as a research tool. You may have recognized phalloidin because you can use phalloidin and make it fluorescent or tag it with other things, and then you can use it using microscopy because you can localize actin. Phalloidin is a very specific actin binding molecule, so if you have a fluorescent phalloidin and you add it to the cell, phalloidin will coat all of the actin, and then you can see where the actin cytoskeleton is inside the cell. Keep in mind, if you're doing that, if you're putting phalloidin into a live cell, first of all, you have to get it into the cell, but then once you stabilized all the actin, that cell is potentially going to die. So normally you don't use these things in terms of, if you're imaging, you're not going to be looking at the kinetics of the actin. If you're imaging with phalloidin, you're likely killing the cell and then just looking where actin was. Okay, so we have, the cell is using a number of proteins to regulate how fast actin can polymerize. Okay, again, actin polymerization is a tool to move things around, so the cell wants to regulate how fast polymerization occurs, and the cell also wants to regulate where polymerization is going to occur. And that's where nucleation comes into it. Okay, we've talked a lot about nucleation, and that, you know, if you have a nucleus, you're going to get to elongation faster. So you have to think of the cell as sort of deciding things, although it doesn't do that. But the cell, whether it's microtubules or actin, it's going to decide where nucleation sites are, and that's where it's going to accumulate things. So if you want actin polymerization to occur in a certain area, you're going to have actin monomers present there, you're going to have profilin present there to make sure they're all in the ATP form, etc. and you're going to have nucleating proteins present there, so that's where polymerization will occur. It's regulating where polymerization is going to occur within the cell, and nucleation is an important part of that, right? If you have a nucleation site, you're going to get to polymerization faster versus if you don't have a nucleation site. So the cell is going to optimize where nucleation is going to occur, and that's where actin can do its job. Okay, so there's a number of ways it can do this. One of them is when you're looking at unbranched filaments. So these are actin microfilaments that are just a straight line. Okay, they don't have any branches. And this involves a nucleating protein called formin. There's a number of different types of formins. Formins are going to regulate what's happening at the plus end. It's going to act as a nucleation site. Okay, so formin is going to regulate where nucleation is. Because polymerization is powerful, that is to say, once actin is polymerizing or depolymerizing, it can move things. You want to regulate formin. So formin is a nucleation site, but it can be regulated by this protein called Rho-GTP. Okay, so often on the plasma membrane or other places in the cell, you'll have Rho-GTP. If Rho-GTP is present in the GTP form, it can activate formin. So in the presence of Rho-GTP, this protein formin can be activated, and then formin can act as a nucleation site. So the little diagrams here of Rho-GTP looking like this, formin looking like this, and there's actin growing at the plus end. Does it actually look like that? It doesn't really matter, artistic license. Up here is sort of a different representation of how formin is working at the plus end to regulate actin nucleation and growth at the plus end. There's a little movie here that may or may not work, right? So formin is at the plus end, and it's going to be acting as a nucleus. Here it doesn't look like it's acting as a nucleus, but at the beginning it acts as a nucleating factor. Once you have nucleation occurring, formin can regulate how fast polymerization is going or how fast depolymerization is occurring, right? Formin is working with Rho-GTP. So if you want it to be polymerizing, you will have the Rho in the GTP form. If you want it to depolymerize, this can go into the GDP form, and you can regulate things. Okay, so formin is regulating what's happening at the plus end. Formin is often activated by Rho-GTP, which is on the plasma membrane. So you can imagine if this is where the formin is the plasma membrane. If that is the plasma membrane, if the actin is polymerizing, you're pushing the plasma membrane up in this situation, and if it's depolymerizing, you're pulling on the membrane. Okay, so if actin is sitting at the membrane, it can push on the membrane, it can pull on the membrane, it can move the membrane. So you have relative movement. Also consider if the membrane is there, but the membrane is not moving. If the membrane is fixed, if the membrane has all kinds of proteins holding it in place, then when you have actin polymerization, the actin filament is going to be extended into the cytoplasm. So if the plus end is growing, but the membrane is not moving, the actin will be pushed down into the cytoplasm. If it's depolymerizing, it will be pulled away from the cytoplasm. Okay, so movement is relative. The plus end can grow or shrink. Either it can push or pull a membrane, or if the membrane isn't moving, then the actin filament itself is moving down or up, depending on if it's polymerizing or depolymerizing. Make sense? So the polymerization and depolymerization can move membranes. Okay, so this is with formin and unbranched actin. The actin microfilament is just a straight line. You can also have polymerization of actin in a branched fashion. So to branch actin, you have to have an existing microfilament. So if you have an existing microfilament, you can grow a branch from the side of it. Okay, so the existing microfilament has a minus and a plus end, and when you grow a branch, you're growing the plus end away from that existing filament. So the plus end is the growing end again. And you have nucleation at the minus end. All right, so you have an existing filament, and you're going to grow a branch, and at that branch point, you need a nucleation site, and that nucleation site is provided by this protein called ARP23. Okay, so ARP23 is going to be activated on an existing microfilament, and then you will have a branch growing away with the plus end growing away. Okay, so ARP23 is a nucleation site. It, even though it is a nucleation site, it actually needs something called a nucleation promoting factor. So up on the top right here is sort of this little diagram of what's going on. It's somewhat complex, but it's hopefully pretty easy. Okay, so you have an existing actin microfilament. You have a minus end and a plus end. You then want to grow a branch. At that branch point, you need ARP23. Right? ARP23, just like formin, needs to be activated. It's going to be activated by two proteins. One of them that's going to be in the GTP form, and one of them that's called the nucleation promoting factor. All right? So there are two nucleation promoting factors and two GTP binding proteins. So there's a nucleation promoting factor called WASP and a nucleation promoting factor called WAVE. And they are activated by two different proteins. One of them is called CDC42 and the other one is called RAC. So CDC42 GTP or RAC GTP. Right, so you have ARP23. ARP23 is at the branch point. It is going to work with either WASP and CDC42. So in this example, here's a WASP protein and the CDC42 protein. They're together going to work with ARP23 to cause branching here. So ARP23 is going to work with WASP with CDC42 or with WAVE and RAC GTP. Make sense? So ARP23 is the protein where the branching is going to occur, and there's two other proteins that need to activate it. It's either WASP with CDC42 or WAVE with RAC GTP, and they both have to be in the GTP form. Okay, so this is going to cause branching to occur. And branching is very common. There's lots of branched actin that forms very complex networks within cells. But this branching is very quick and rapid. And this branching is actually, an example of how quick and rapid this branching is seen in a situation involving a parasite called Listeria. So Listeria is a parasite that gets into cells, and then Listeria has proteins on its surface that actually activate ARP23. So Listeria kind of circumvents all this WASP and CDC42 sort of business, and it directly activates ARP23, so you have lots of branching going on. So right next to the Listeria, you have these branching actin molecules, and this rapid branching pushes the Listeria around within the cell, and ideally it's pushing the Listeria so fast that it pushes it through the plasma membrane into the next cell so it can infect multiple cells. So there's a little movie here, hopefully works, that shows you this, where these little black dots are different Listeria pathogens inside the cell, and part of that Listeria is activating ARP23. So you can see all these little cloudy bits behind all these Listeria. That's actin being rapidly polymerized because of ARP23 activation. And you'll see all these parasites being pushed around the cell, and you'll see a lot of them smack into the plasma membrane. And there is no other cell next to this, but potentially it can be infecting other cells that burst through one plasma membrane into the next cell. Let's see if this movie works. Okay, so this is time-lapse, but still, these parasites are moving very quickly, and this movement is occurring because of rapid ARP23 polymerization of branched actin. So actin polymerization is very powerful. It can move things. It can move parasites. It moves membranes. So that's why it's very well regulated. Okay, it's regulated in unbranched actin by formin, and it's regulated formin working with ROGTP, and it's regulated with branched actin using either WASP with CDC42 or WAVE with RAC. Right? Okay, so actin polymerization can move membranes. So by moving membranes, you can do lots of different things. And in your mind, some of this may be difficult to picture, but just appreciate movement is relative. If the actin is standing still and polymerizing, it can push on things or pull on things. If whatever the actin is pushing or pulling on, the actin will move. So even in your mind, if you can't picture how is this working, just appreciate actin can pull or pull on, can push or pull on membranes, or the actin itself moves. So actin is critical in endocytosis, right? So you have a lot of processes that Dr. Cumming talked about, how you get endocytic vesicles, etc. But one of the things is you have to take that membrane and make a little vesicle with that membrane and then move that vesicle into the cytoplasm. And this involves actin polymerization and involves ARP2/3 activation and then binding the membrane to this growing actin microfilament. So in the place where endocytosis is going to occur, you're going to get polymerization of actin, branching of actin. It's going to grab onto that membrane and then grab that vesicle and then move it into the cytoplasm. So as you're polymerizing actin at the plus end here, you're going to be moving this vesicle deeper into the cytoplasm. So endocytosis involves actin polymerization, involves ARP2/3 activation, and you're pulling on the plasma membrane, and eventually the vesicle is going to be pushed and moved throughout the cytoplasm. You can do this experimentally. On the right-hand side, you have in red are endocytic vesicles, and you can, just like with the listeria experiment, if you kind of polymerize actin behind the endocytic vesicles, you can move the endocytic vesicles around, which is sort of what's happening here, except you're pushing along against the plasma membrane here as well. So in your mind, if you can't picture exactly how is that moving, what's moving with respect to what, it's okay, just appreciate that actin polymerization can pull on a plasma membrane, and then once you form a vesicle, it can move that vesicle into the cytoplasm. Okay, so here actin is pulling on a membrane. Actin can also push on a membrane. We'll talk about this later on when we talk about cell migration, but another example here is phagocytosis. So you have a cell, and it has some sort of pathogen, and this cell, the white blood cell leukocyte in this case, is going to phagocytose this pathogen. And it does so by polymerizing actin. So it has receptors there, it recognizes this pathogen. It's going to trigger actin polymerization. Actin is going to polymerize, it's going to push the membrane forward, it's going to form these protrusions of the cell, and eventually it's going to encompass this pathogen through phagocytosis and then degrade it later on. Okay, take-home message in the last couple of slides is actin polymerization. Yes, you need other proteins to bind the plasma membrane to push and pull on things, but actin polymerization by itself can push or pull membranes and vesicles and things. Make sense? All right. So, but to do this, actin has to be bound to these membranes and things. So there's a number of proteins that are regulating what actin can do that allow it to do these functions. So actin can form bundles or actin can form networks. So actin bundles are basically straight, unbranched microfilaments that are bound together to kind of form these structures called vertical structures. So actin can form bundles or it can form networks. Networks are branched actin, but even branched actin can be further cross-linked by other proteins. Okay, so there's 4 proteins I'd like you to become familiar with. There's fibrin and alpha-actinin. Both of these proteins take parallel actin microfilaments and bundle them into structures, actin bundles. Right, so fibrin and alpha-actinin are involved in bundling of unbranched actin microfilaments. And a protein called spectrin and filamin are involved in making networks. So they can take already branched actin or unbranched actin, it doesn't matter. Spectrin and filamin are going to form complex networks. They cross-link, so things are not parallel anymore. Okay, so you have 2 proteins that are involved in making bundles, so not branched, and 2 proteins that are involved in branching structures and forming networks. Straightforward? Okay, a lot of this is occurring at the cortex. If you remember, one of the things we described about actin is that it's cortical. So actin is found underneath the plasma membrane. And it's often involved in moving the plasma membrane and supporting the plasma membrane. Right? I don't know if I've said it before, but certainly I'll say it several times. A phospholipid bilayer, the plasma membrane is not strong. A phospholipid bilayer is a good barrier. It has sort of semi-permeability that's important, but it's not a strong structure. So if you want strength to the plasma membrane, it has to come through proteins. And some of those proteins are held in place by actin. So you have cortical actin. There's actin found underneath all our plasma membranes, basically, and the actin is linked to the plasma membrane. So there's lots of proteins that are interacting with the plasma membrane that then interact with the cytoskeleton, be it actin or microtubules or intermediate filaments. In this case, we're talking about actin first. Actin is cortical. It's found right underneath the plasma membrane. Most of what's supporting the plasma membrane and involved in movement of the plasma membrane involves actin. So if you think of a red blood cell has that nice biconcave shape. That shape is present because when it goes through capillaries, it can deform and it can kind of get abused. So a red blood cell plasma membrane is actually quite strong, and the reason it's strong is because underneath its plasma membrane, it has this complex actin network that holds things in place. So this is an electron micrograph of the inside of a plasma membrane of a red blood cell, and this is sort of a schematic. So the plasma membrane itself, the phospholipid bilayer, isn't that strong, but within it you have lots of transmembrane proteins, and we'll talk about those transmembrane proteins later, or at least talk about transmembrane proteins. And all of those transmembrane proteins are linked together with actin and other actin-binding proteins such as spectrin, which we just talked about. So if you remember, spectrin can work with actin to form networks. So underneath plasma membranes, in this case it's the red blood cell plasma membrane, you have this complex network of actin and other proteins such as spectrin, and those proteins are bound to the plasma membrane, supporting that plasma membrane. An important protein here is called ancrin. Okay? So ancrin works with this network of the actin cytoskeleton to support the plasma membrane. So ancrin is anchoring the actin cytoskeleton to the plasma membrane. One of the proteins that does that. There's other proteins. So ancrin is working with actin networks. In other cells, you have actin bundles. For example, there's these structures called microvilli. Microvilli are present on lots of cells, and they're used to increase surface area. So a lot of your epithelial cells in your gut that absorb things have microvilli. Microvilli are projections of actin bundles. Okay? Do not confuse microvilli and cilia. Cilia are motile, and they contain axonines, which are microtubules. Microvilli are not motile. They contain actin bundles. So these actin bundles would be held together by those proteins we just talked about. There's fibrin and alpha-actinin that are holding these actins together. But you also want to hold this actin to the plasma membrane. Right, so this actin is held to the plasma membrane by a number of proteins. In this case, there's a protein called esrin. So in some cells, this protein called esrin is linking actin bundles to the plasma membrane. So you can link actin bundles to the plasma membrane through esrin. You can link actin networks to the plasma membrane through ancrin. So far, so good? Other times, you want to link the cytoskeleton through strong sort of physical proteins. So that's the case in muscle. Inside muscle cells, you have these structures called sarcomeres, which hopefully we'll get to this lecture. So I think you guys have seen sarcomeres in other classes before. Sarcomeres contract and muscles move things. So the sarcomere is inside your cell. it's going to move things outside of your cell. Normally, it's attached to something like a tendon. So you have a tendon outside of the cell, and you have a sarcomere, which is actin and myosin, inside of the cell. So outside of the cell, you have extracellular matrix, such as a tendon. Inside of the cell, you have actin and myosin that are going to provide contraction. So when you want to move, your actin and myosin move, and they need to move that tendon so that tendon can move a bone, and then you flex your elbow or whatever. But you need a connection between the cytoskeleton, that actin and myosin that's inside the cell, and then the tendon or the extracellular matrix that's outside of the cell. And for that, you need actin binding proteins and transmembrane molecules. So there's lots of transmembrane molecules, and we'll talk about some later on, that link the extracellular matrix to the cytoskeleton. So the extracellular matrix are proteins that are found outside of cells. Collagen is one that probably everyone's heard of. Your tendons are made of collagen. There are proteins that are binding to the collagen, proteins that bind to the plasma membrane, but then you have to link those proteins to the actin cytoskeleton, which is actually doing all the movement. One of those proteins is called dystrophin. There's a huge complex here. There's lots of different complexes that involve dystrophin. But normally, when a normal muscle cell contracts, you have the actin that's moving, and we'll talk about that when we talk about muscle contracting in a second. The actin is going to pull on this dystrophin protein, that's going to pull on this transmembrane molecule, that's going to pull on this extracellular matrix, which is the tendon, that's going to pull on your joint. So you need this connection. There's lots of diseases called muscular dystrophy, and a lot of them involve sort of some sort of defect in this sort of complex, right? The simplest one, I think, is Duchenne's muscular dystrophy, which is a mutation in the dystrophin itself. So if this dystrophin protein isn't working, every time the actin and myosin pull, it doesn't properly pull on the tendon. So the muscle keeps on trying to pull, but this protein, the dystrophin, is not working properly and not transmitting this force, and the muscle cell keeps damaging itself because it can't pull on the tendon. So the muscle cells are repeatedly damaging themselves, and it's a progressive thing, and after years of damage, it becomes fatal. Okay? So the actin binding protein dystrophin is involved in sort of physical support in muscle cells. It's involved in other cells as well, but in this case, the damage is quite severe. Okay, so you have ankrin involved in binding networks to plasma membranes. You have ezrin involved in binding bundles to plasma membranes, and a protein such as dystrophin, which is very specific in binding the actin cytoskeleton to the extracellular material outside of muscle cells, as examples. And we'll talk about muscle cells hopefully very soon. All right, so actin by itself can do lots just by polymerizing and depolymerizing. And it can even do more because it has motor proteins. Okay? Motor protein of actin is myosin. So myosin is a plus N directed motor. There basically is no minus N directed motor with actin. There are many types of myosin. We're only going to talk about 3 of them, and that's on the next slide. So again, with myosin, as we talked about with kinesin and dynein, it sort of has the same structures. And let's just kind of stick to the simple things, right? So myosin has heavy chains and light chains. The heavy chain has a head domain, a neck domain, and a tail domain, okay? So that part is kind of the same. You have a heavy chain that has a head, neck, and tail. The head domain is the one that's going to bind to the actin. The head domain has ATPase activity. The head domain is going to move to the plus N. Okay, so heavy chains, head, neck, and tail. It has light chains, but the light chains are not involved in cargo. The light chains are involved in binding the neck region, and they're involved in regulating how the myosin moves. So the light chains are basically regulatory. They regulate how fast the myosin will move. Okay, myosin is very abundant. It's in all cells. It's certainly very abundant in muscle cells. So when people were starting to look at myosin function, you can isolate myosin heavy chains. You can isolate myosin heavy chains. In your muscle, the heavy chains look like this. It's called the fixed filament. We'll talk about it on the next slide. But you can isolate lots of myosin from muscle, and then you can take that isolated myosin and degrade it with specific proteases. And by degrading it with specific proteases, you get different fragments. And then therefore you can study what those different fragments do. So you don't need to memorize any of this other than appreciating this is how we discovered S1 myosin, which is basically the head domain. So you can use a protease and you can cleave off the tail domain, and then therefore you can ask, well, what does the tail domain do? Because without the tail domain, you may or may not be able to do things. And eventually you use different proteases and you get to this S1 myosin, which is basically just the head domain. And you know that that head domain has ATPase binding activity, the head domain can bind to actin. And we know if you add the S1 myosin to an existing actin microfilament, the head domain will bind to it, and it will decorate it, and it will make that arrowhead that points to the minus end. So that's what that S1 myosin is. It's just a proteolytic product of cleaved myosin that's basically the head domain of the heavy chain. Okay, so you have heavy chains and you have light chains, and there's different types of myosin. Okay, 3 of them are all we need to memorize. Let's start off at the bottom here with myosin 5, because in many ways that's the easiest one to understand. Okay, so myosin 5 has 2 heavy chains and a number of light chains. So it has 2 heavy chains where the tails are bound together, and myosin 5 is involved in organelle transport that binds the cargo. So myosin 5, like kinesin, the head domain is going to bind to the actin here, the head domain is going to use ATP, and it's going to move to the plus end. The tail of this heavy chain is going to bind to the cargo. So there's no light chains involved in cargo binding. The tail domain has specific sort of moieties, and it's the actual tail of the heavy chain that's bound to the cargo. So myosin 5 is moving cargo, but it's using the heavy chain tails. Myosin 5 has light chains. These light chains are on the neck region, has 6 light chains, and those light chains regulate its movement. And myosin 5, we'll see in a second, is the fastest moving myosin because all these light chains allow it to move quickly. Okay, myosin 5 involved in organelle transport, 2 heavy chains, and it's the tail of the heavy chain that binds to the cargo. The light chains are regulating its movement. Let's skip up to myosin 1. Myosin 1 is a little unusual in that it only works as 1 heavy chain. Right? It doesn't work as a pair. All other ones are multiple heavy chains. Myosin 1 works alone as a heavy chain. It does have a number of light chains bound to it. Myosin 1, though, has a unique property in that it has a short tail, and that tail can bind to membranes. So myosin 1 can bind the actin cytoskeleton to a membrane, again, supporting membranes. One of the jobs of actin is to support membranes, and here myosin 1 can do it. It is a motor protein. It can bind to a membrane. It can move to the plus end, but it doesn't have to. If it doesn't move, it's just linking the plasma membrane to the actin. So in all these cases, you have these motor proteins, but if they don't move, they're just holding things in place. Okay, so myosin 1 is just a single heavy chain. It can link actin to the plasma membrane. It can move to the plus end, but it doesn't have to. All right, now we come to myosin 2. Myosin 2 forms a homodimer, so you have 2 heavy chains coming together, but then you have a number of heavy chains that make something called a myosin 2 fixed filament. So just like kinesin 5, if you remember, kinesin 5 had head domains on both sides, except there was only 4 heavy chains for kinesin 5. There's 2 head domains on one side, 2 head domains on the other side for kinesin 5. With myosin 2, there's many heavy chains that come together to form something called the thick filament. So there's lots of head domains on one side, lots of head domains on the other side, and a lot of overlapping tails in the middle. So you have a myosin 2 thick filament of many myosin heavy chains that come together, and these myosin thick filaments are often found in muscle in between actin microfilaments. And they're involved in contraction inside the sarcomere, which we'll hopefully see in a little bit. Okay, so myosin 1 can bind to plasma membranes and it can hold them. Myosin 2 can form thick filaments and it's often found in muscle contraction. And myosin 5 is the most rapid moving one and it can be, it's found in organelle transport. Okay, so you can do lots of different studies to kind of show the different properties of it, especially the length of the neck region and the presence of these light chains. So there's several studies you can do. A lot of them involve something called the sliding filament assay. So you can take fragments of myosin. Remember, I just described you can isolate myosin heavy chains and you can degrade different fractions of them and study what different parts do. So we can have different fragments of myosin and bind them to something like a cover slip. So you can have the head domain and the neck domain, or the head domain, the neck domain, and the tail domain. You can have different fragments, different lengths of things bound there, and then you can add actin and you can try to see how the actin moves. And then you can measure the step size or the speed. So again, hopefully there's a movie that works here. Hopefully it actually has sound. Give me a second here. Let's see if this works.
Is the sliding filament assay. First, myosin molecules are added to a chamber formed between a cover slip and glass slime. Some of the myosins will adhere to either the cover slip or the slime. A solution of BSA, bovine serum albumin, is used to wash away the excess myosin molecules and block all unbound sites in the chamber. Next, a solution of fluorescently labeled actin filaments is flowed into the chamber. A closer look inside the chamber reveals actin filaments bound to a proportion of the myosin molecules. In the presence of ATP, a myosin head is activated to walk towards the plus end of an actin filament. Since the myosin molecule is tethered to the glass slide, the resulting action is sliding of the filament, with the minus end leading. The sliding movement of an individual filament can be visualized under a fluorescence light microscope.
So that's what they're trying to show you in these little images. You have different fluorescent actin, and then you can look at how fast things move or what the step sizes are. So by putting on different fragments of myosin under these tests, you can look at to see what does what. So for example, we know that the length of the neck plays a role in speed. So this is with myosin 2. The longer the neck is, the faster the myosin can move, and that's because it can bind to more light chains. So the longer the neck, more light chains, the faster you can go in general. And the master of that, or the champion of that, is myosin 5. Myosin 5 has a long neck. It has 6 light chains on each neck, and it has the largest step size. It has a step size of 72 nanometers. So again, to me, the step size is it's one head moving over the other head. So one head is moving past the other head, and that's 72 nanometers. And if you remember, 72 nanometers is the size, is the turn for the one length of the helix, for the actin double helix that it's made. Okay, so between the two heads, if you're not moving, the two heads are 36 nanometers apart, but each head is going to move 72 nanometers with each step. Okay, so as you saw previously, there are different step sizes. You don't need to memorize these, but appreciate that not all myosins are moving at the same step size, and that's largely due to the differences in their neck region, and that's because of the light chains that bind there. All right, so how do they move? So we're going to talk about how myosin uses actin to move along, myosin uses ATP to move along actin. So I'm going to describe it, we're going to watch a movie, and I'm going to describe it again, and then everyone will get it. So what do we have here? So this is sort of a repeating thing where you have a myosin thick filament. So if you remember, a myosin thick filament is made of myosin 2. All the tails overlap, and on both sides are head domains. So this is trying to show you the head domains from this thick filament coming down and reaching actin. So this is actin, and you have a thick filament, and then that's sort of repeated a bunch of times. Okay? So you have myosin head domains coming from this thick filament, and they're going to bind to the actin. So muscle contraction is cyclic, right? Here we are starting off in a state called rigor. Okay, so muscle contraction doesn't start in rigor state. My explanation is starting in the rigor state. Okay, so there is no real start, it's a cycle. But we're going to start off in something called rigor. What is that? That is the head domain is bound to actin, and it's in the ADP form. So myosin is bound to actin, and the myosin is bound to ADP. And it will stay in that rigor state indefinitely. Myosin is quite happy, bound to actin, and it's in the ADP form. It will stay bound to that actin. So nothing happens until you replace that ADP with an ATP. So when ATP binds to myosin, the myosin lets go of the actin. So ATP binding causes release. Once there's release, there is hydrolysis of that ATP. So that ATP is hydrolyzed, but the phosphate is not released. So inside the myosin is ADPPI, the phosphate is still present. So the hydrolysis of the ADP causes a conformational change where the myosin will bend its neck and bind to an actin towards the plus side. Okay, so ATP binding causes release. ATP hydrolysis causes a conformational change that the neck bends and you bind to an actin towards the plus side. The step size is going to depend on the neck and the light chain. Okay, once it binds to that, so far there is no relative motion between the actin and the myosin. All you've done is let go of one spot and you've moved to another spot. So the actin and myosin have not moved relatively. But once the myosin binds to that new site, the phosphate is released. Once the phosphate is released, now we have something called the power stroke, where the myosin will bend its neck and move its head, and that's where you get the relative movement of the actin to the myosin. Okay? So when you release that phosphate, the power stroke occurs, and you're back where you started. Now you have myosin in the ADP form, again, that's down to the actin. So when you're back to rigor state. All right, let's go for the exciting movie.
Myosin motility is coupled to ATP hydrolysis. The following mechanism pertains to myosin 2, but all myosins are thought to move the same way. Exchange of ADP for ATP opens the cleft, disrupting the actin binding site. This causes myosin to release the actin filament. With ATP hydrolysis, a conformational chain rotates the myosin neck and partially closes the cleft, restoring the actin binding site. Myosin rebinding to actin catalyzes phosphate release and activation of the power stroke. The final step of the cycle is ADP release and binding of a new molecule of ATP. Movement of myosin along the actin filament continues as the cycle is repeated. In this case, the myosin molecule is tethered, so the resulting action is sliding of the actin filament.
Okay, that movie, or at least a newer version of that movie, is on the Achieve website, and I think the other one is as well. Okay, so Again, you have actin and myosin, and there's going to be relative movement, and that relative movement occurs during phosphate release, right? So prior to that, there is no movement. So ATP binding causes release of the myosin head from the actin. ATP hydrolysis causes movement of that head towards the plus side and binding to a new actin. And it's the release of the phosphate that causes the power stroke, and then there's relative movement. And with the release of that phosphate, and after the power stroke, you're back in rigor state, which is the myosin bound to ATP, happily bound to the actin. Okay, so rigor, you probably couldn't figure out, rigor mortis happens after death. So after death, you run out of ATP in principle, and that happens whatever 8 to 10 hours depending on temperature and stuff. So rigor mortis is that after death, you have no more ATP. So your myosin binds to your actin, and it just sits there, and all your muscles are stiff for a number of hours until things start to degrade. So rigor mortis occurs because you have no more ATP. Okay, so it's part of a cycle. Okay, so this cycle is involving ATP. So this is overlapped with another cycle involving calcium, and you guys have to get those straight in your mind, hopefully. But hopefully this part makes sense. Okay, so let's get into muscle contraction. So sarcomere, I think you guys have seen pictures like this in high school and other bits and pieces. And hopefully this will begin to make sense, but let's kind of go over what we have here. So a sarcomere is this repeating structure that's found in striated muscle. So other muscles have similar arrangements. But basically what you have is actin and myosin in such a configuration that when the actin and myosin move, you're contracting, you're causing a shrinkage. So when you look at a sarcomere, it's an arrangement of actin microfilaments that have myosin thick filaments in between them. So you have these structures called Z discs, or Z discs, I guess if you're American. From these Z discs, you have these projections of actin, like these actin fingers coming out, and in between them you have myosin thick filaments. So when you're looking at this sort of arrangement, you can, a couple things. One is, you can give names to these things. So there's lots of different names that are associated with sarcomeres. We're only going to stick with 3 things, right? The A band. The A band is basically the area of the thick filaments. So the A band represents the thick filaments. The I band represents the space between the thick filament and the Z disc. So there's I bands on both sides, and this is a repeating structure, so there's I band on either side of the Z disc. Right, so the A band is the thick filament, the I band is the space between the thick filament and the Z disc. There's other bands and other anatomical things, we're not going to deal with it. When you look at the arrangement of the actin, you can see that the plus end of the actin is found at the Z disc. The minus end of the actin is found in the middle of the sarcomere. In between all these actins are these myosin thick filaments. Myosin is a plus end directed motor. It is going to want to move to those plus ends, and then indeed that's what happens during contraction. So you have these thick filaments binding to these actins on both sides. So when you have the head domains of the thick filament on one side trying to get to the Z disc on the left, and you have the head domain on the other side trying to get to the Z disc on the right, what's going to happen is that the actin is going to move in a relative way such that you get a decrease in the spacing between the Z discs. So the Z discs are going to be brought closer together. So during muscle contractions, the Z discs come closer together. The I band decreases in size. So the I band is the space between the thick filament and the Z disc. But the A band does not change in size. The A band is the thick filament. It doesn't change in size. It's just pulling things around it. So the I band decreases, Z discs come closer together, the A band does not change. Okay? It's a little more complex than that. There's a few more proteins that are involved in stability. You do not want actin polymerizing and depolymerizing inside your muscle cell. So there's lots of proteins involved in stability. The two ones that are the most obvious one is cap Z at the plus end and tropomodulin at the minus end. We already talked about those capping proteins, and here they are here. Cap Z is at the Z disc, that's why it's called the Z disc, because cap Z is there. And then tropomodulin is at the minus end. So the actin is not going to polymerize or depolymerize, and that's what you want, and that's where we'll stop, and we'll start with this slide again next time.
Because I'm curious, once they construction, how can they?
That's next one. That's next one.
So I asked.
Yep.
Okay.
So when you mentioned like the calcium, is that talking about the calcium thing?
That's correct. We'll get to that next time.
Thank you.
Well, so if you go back to that movie, I don't know if you have the movie yet. So if you depolymerize the plus end, if you have form in there, you will get your full. Right, so if the microfilament is shrinking, but you're still attached to the plasma membrane, you're full. Yeah. It's growing, it's pushing. It's polymerizing it from push, it's depolymerizing it from pull. If you're attached to it, yes. Wow. Sorry.